Good morning, students. This is Prasad, and introducing myself as my name is Prasad, where I having a 15 years an experience of teaching is presenting in the biology, and today we are going discussing the 10th class of an ICSE, a board that is a genetics. So here coming to the genetics, here genetics means here will consisting of the two words are presenting. So here what are the two words means here it consists of one is heredity, and second one is the variations. So these are the two words where it is presenting in an genetics or heredity. Here otherwise heredity we can use the word as an inheritance also. Inheritance. So here what is a heredity means or inheritance? Here the heredity or otherwise we can use the word as an inheritance. Inheritance means the transmitting of characters, the transmission of the characters from one generation to another generation, from one generation to the another generation. So the transmitting of characters from one generation to another generation we can call as an heredity, or we can call as an inheritance. And here variations. What are variations means the variations. Here variation means the characters, the individual characters which are present in an organism, we can call them as the variations. Here the individual characters, individual characters which are present in an organism. So here in an organism, so we can call them as the variation. That means individual character, that means the characters which are presenting. So that characters which are transferred from generation to generation, we can use the word as the heredity, or we can call as inheritance. But variations, the characters, individual characters which are presenting, they are called as variations. Here the variations, if it taken place in a twins, example, here example if it taken place a twins, here twins, they are morphologically, they look like the same only, but when coming to the characters are differ from one to the another. So that the differential characters which are presenting in an individual organism, then we can call them as the variations. So if it taken place a twins also, they are morphologically look like the same only. They are morphologically look like same, but coming to the internal characters, here the internal characters, one is differ from the one person, that is in twins also they are one individual is differ from the an individual, then we can call that as the variations. So here the variations will be taken place in two types. Here this may be taken place in two types. One is we can call as somatic variations. Somatic variations. And second one is the genetical or germ variations. Genetical variations or germ variations. So here the somatic variations. Here the somatic variations which are presenting here, these are presenting in the autosomal chromosomes that are present in the somatic body cell that are present in the autosomes, where here autosomes means here we know that the chromosomes are classified into two types. Here the chromosomes are classified autosomal chromosomes and the sex chromosomes. There are autosomal means here first one is somatic chromosomes or somatic variations. Somatic the variations. These are present in an autosome. Autosomes, or otherwise we can call as the body chromosomes also, body cells or body chromosomes. So here these are autosomes which are present in the somatic body, somatic cells of a body. We can call them as somatic variations. So here these somatic variations does not have an important, they does not place no importance in variations. They does not place any important role in the variations. And second one, if it is this germ or genetical variations, So here these are present in the sex chromosomes. These are present in the sex chromosomes. These are present in sex chromosomes, or otherwise they are called as the allosomes, where here these sex chromosomes will carry the variations. There means these sex chromosomes will transfer from one generation to the another generation. So here they place an important role, role in variations. Why means here these only will helpful for producing of the characters, new characters or individual differences characters are produced by these germ or genetical variations where they are present in sex chromosomes, or we can call them as allosomes. They plays a very important role where here new characters are produced in an individuals. So that is where we can take place in the variations, two type of variations. One is somatic and second one is the genetical variations. Now coming to the here the way genetics, it was explained by a scientist called as Gregor John Mendel. Here the Gregor John Mendel Mendel he is a scientist where he experimented in heredity and also the variations. Here he experimented on heredity as well as the variations. He conducted the experiment in a plant called as pea plant. Pea plant, that pea plant is called as Pisum sativum. Pisum sativum, it is this the scientific name of the pea plant where he is taken place the experiment, and he is born on 1822 in an Australian monk. So here he is an Australian monk, but here in 1956, in the year 1856, he have given experimental proofs of the heredity. That means he taken in 1856 he conducted the experiments on the Pisum sativum, and here what he conducted the experiments on Pisum sativum. Why he selected the plant of the Pisum sativum means here they can easily first one. Why means why he taken place the Pisum sativum or a pea plant means here the cultivation is very easy. Cultivated of plants. Cultivated plants are easy, and also here the time period is very short. That means lifespan is very short, and also here in a small area he can grow the plants. And also here fourth one is he can able to cross by self breeding or cross breeding or cross pollination can be taken place. He conducted experiment, that is cultivated the plants by the cultivated plants by the hybrids, by hybrid by the self pollination, by the self pollination or cross pollination. That means here he can able to done the experiment by the self pollination or a cross pollination can be taken place by this. So these are the easy characters where he can having with the help of these characters we have selected the Pisum sativum. And also here will having here he taken place the seven pairs of the contrasting characters. He have taken place the 7 pairs of contrasting characters are taken place. That is 7 contrasting characters are first one is the position of the flower, position to the, that means position or the color or otherwise the shape of the pod or the color of the pod. So such characters here taken place. Here he taken place the 7 pairs of contrasting characters are taken place where he conducted the experiment in 1866. In 1866 here he conducted. That means this was his experiments was not approved by the Born Society of History. Here Born Society Society of History is not accepted by his experiments. By means he given a mathematical calculations are taken place, but they are he inverted the experiments. What he conducted the experiment that are felt on a deaf ears. That means the scientists which are approved, they are not be approved by the scientist, that is the Born Society of the History. It is not accepted. But later in 19th century, in 19th century, here they experimentally, the three scientists of European scientists, the scientists, again they conducted experimentally by an individual. They experimented. The individual experiments are taken place, such as the Hugo de Vries in Holland, and Testmark as well as that is coherence. These are the three sentences. One is the Holland, Australian as well as the German. So these are the three scientists where they conduct the experiments. One is Hugo de Vries, Testmark and Horens. But what he conducted individually, that was already proved by the Mendel. Then his work, that means Mendel work was approved, and they approved by the Natural Society, that is Born Society of Natural Society is accepted, where they are conducted by individually by Hugo de Vries, Testmark and Horens. So here we can call them as the Mendel. We can call them as already they have inverted by the egregore John Mendel. So here is called as the father of experimental genetics. The father of an experimental genetics. So this is where that is taken place by the egregore John Mendel, where he experiment the Mendel in the heredity, in the form of heredity as well as the variations. So he conducted the experiments on the epiplant, that is called as Episum sativum. Now coming to the next one. So here coming to the basic genetical terms, basic genetic terms. So here coming to the basic genetic terms. So here first one is a gene. So what is a gene means? It is a fundamental character of an That means it is a fundamental character of an organism. So it is called as a gene. It is a basic, a fundamental character present in an organism that we can call as a gene. Where here Mendel used this gene as Mendel used this as a factors. Use the word as the factors. Factors means Mendel is not used as a gene. The word gene was given by Johnson. So here the word gene is given by Johnson, but here Mendel not used the word gene, used the word as a factor. There are some factors which helpful for the formation of a characters in an organism. They are called as factors, but they are not factors given the word as a gene. It is a fundamental character of an organism. Next, second one, the alleles. Here it can be defined as the alternate form of a gene. It can be the alternate form of a gene present in a locus of a homologous chromosome. Homologous chromosome is called as a gene. That is, it is the alternate. It can be defined as the alternate form of a gene present in an locus at a point of in a locus of a homologous chromosome. We can call as a gene. So here example alleles means we can take in place the alternate form means example tall and a draft. So here these are the tall is a one gene and draft is another gene where they are present at a point called as the alleles. That is alternate form of a gene which is presenting we can call them as the alleles. Next, if you see in the phenotype. Here what is the phenotype means? Here it can be defined as the external structure of a gene. The external structure or the external characteristic feature of a gene is called as a genotype. Here an external characteristic feature of a gene is called as a phenotype. Here the phenotype means here external character, that means showing the outer form of an external character which is presenting, then it is called as a genotype. Here genotype means, for example, if you see a black and black colored as well as the light colored. So here external forms where externally, that means outside characters, what are the outside characters are present on a gene, we can call them as a genotype. Next, fourth one, that is phenotype and genotype. Here genotype means here it is alternate is internal structure, internal characteristic feature, feature of a gene, or we can write this is an internal constituents of a gene, we can call as a genotype. So here internally it showing, so that internal structure which is showing we can call as the genotype. Next, coming to the so remaining we'll discuss in the next class. That will remaining we'll discuss in the next class.